Synthetic biology is an emerging technology that is prone to revolutionize the way we understand and interact with nature. Located in the Spanish National Center of Biotechnology, Madrid, the laboratory led by Victor de Lorenzo is an active research group in the field of synthetic and systems biology applied to bioremediation. They are committed to understanding how bacteria sense the environment through transcriptional and metabolic genetic circuits. Ultimately, their goal is to translate this information into biotechnological applications, such as the treatment of polluted ecosystems or the detection of anti-personnel mines in war zones.
Our laboratory has been interested for a long time in exploiting the wealth of biological activities that one can find in environmental bacteria for a suite of biotechnological applications. In the late 70s, recombinant DNA technology was made available to biotechnologists, and that supposed a quantum leap in terms of the possibilities that we had to train and to coach and to domesticate bacteria for various biotechnological objectives. But the big revolution is happening right now because of the onset of systems and synthetic biology that allow us to program and to engineer and to refactor naturally occurring microbes with new properties, and we can now design them for specific purposes with a degree of predictability that was simply impossible to get through traditional genetic engineering.
Bacteria are a remarkable example of adaptation to the most diverse types of ecosystems. This ability resides in the catalog of genes and how these genes interact to sense external conditions, adapting their metabolism and physiology to an ever-changing environment.
We seek to understand the mechanisms of how bacteria sense and process environmental information through diverse genetic networks. That's why we have chosen Pseudomonas putida, which is a unique species of soil bacteria that is easy to manipulate in the lab.
Pseudomonas putida is a gram-negative bacterium with a very diverse metabolism for the degradation of organic pollutants, thus holding a great potential for detoxifying contaminated areas due to different human and industrial activities. Since Pseudomonas putida is a non-pathogenic bacterium that can be safely released into the environment, it is widely used in bioremediation laboratories around the world.
Within a given bacterial population, which is genetically identical, different groups of cells have different, sometimes opposed, metabolic behaviors. This decreases the efficiency of biotechnological processes. So it is important that we study bacteria at single cell level so that we can modify this problem.
We use a variety of novel techniques to study gene expression with fluorescent proteins that are stably integrated into bacterial chromosomes using two types of vectors that we have developed in our lab. These vectors are based on the TN5 and TN7 transposons. Transposons are DNA mobile elements. We then resort to a combination of flow cytometry, time-lapse microscopy, and Raman and optical tweezers to assess gene expression at the single cell level.
Another challenge that we face in the study of bacterial metabolism is the lack of an accurate language for the description of the underlying genetic networks. Whereas biological science describes phenomena in a not very clear way, engineers use a standard language based on precise rules that makes it evident and understandable. That's why systems and synthetic biology use the engineering approach to normalize and standardize biological information.
The element that distinguishes systems and synthetic biology from traditional molecular biology is the focus on the description, redesign, and construction of the core components in organisms. The new paradigm of this approach is that living systems are composed of different parts with independent functions, such as, for example, sequences of DNA like a gene or a promoter. These parts are arranged into biological devices to perform specific roles, and just like transistors and resistances are wired together in a radio, the connections between these parts can be expressed through logic gates, the standard language used in electronics.
Pseudomonas putida contains a very well-organized set of genes, all very connected in a very specific way that responds to environmental signals. Here, using a combination of experiments and mathematical modeling, we have described that pathway in terms of its logic behavior, as for instance, in studying the dynamic range of the signal across the circuit, as if it were the voltage of electronic circuits. This way, we can actually rewire the connectivity of the pathway in order to look for novel functions or to use new components that we can plug in as if we were handling electronic devices.
The extensive understanding of a bacterial metabolic route has allowed a very efficient modification of its mechanism for biotechnological purposes.
By using transposon-based tools, we can engineer genetic circuits that detect pollution from toxic aromatic compounds such as xylene, toluene, benzoate, and other hydrocarbons, which are released to the environment as a consequence of different human and industrial activities. Bacteria expressing these novel circuits are able to respond to these toxic chemicals by emitting light, so they actually behave as living sensors.
This system has also been engineered to develop a very promising tool. Through novel techniques such as mutation-prone PCR and DNA sequence shuffling, the pathway has been modified so that it responds to 2,4-dinitrotoluene, a volatile component from anti-personnel mines. Now, Pseudomonas are able to detect the presence of this compound in minefields and could be used as a tool to facilitate mine clearance.
For the detection of other pollutants, we have developed a strategy using recombinant antibodies from camels. These antibodies are small molecules that can be easily generated in Pseudomonas to detect enzymes from bacteria that grow in contaminated environments. This way, we can assess the health of ecosystems.
But bacteria are not only able to work as biosensors for toxic chemicals, they also possess an extraordinary ability to detoxify the environment.
In the lab, we have constructed a Pseudomonas putida strain that is able to degrade the contaminant 1,3-dichloropropene. In order to do this, we have recruited genes from 3 different bacterial sources, and we have put them together using synthetic biology techniques and introduced these constructs into P. putida. And now the engineering bacterium is able to degrade this pollutant under completely anoxic condition, something that was not possible before the manipulations.
The worst types of pollutants are those that cannot be biodegraded, such as heavy metals and metalloids. The presence of these chemical compounds poses a huge environmental problem that can be tackled through research.
One possibility that we have explored in collaboration with a Chinese group and an American group is to take a gene from a given organism that is able to convert inorganic arsenic, that is one of the most, vicious forms of arsenic, into another arsenic species that is still arsenic, but is not as bad as the former form. And by manipulating the genes and taking the genes from one microbe and put them in Pseudomonas putida, creating strains that now we can put back into the environment, back into soil, and convert this bad form of arsenic into something that is far more innocuous.
In the final challenge of preparing bacteria for environmental release, Victor's lab has validated a system based on gelatin capsules for the preservation of Pseudomonas putida in soil. This preparation allows engineered bacteria to be functional in nature. Hence, they can be used as efficient tools for bioremediation.
For the last 100 years, chemical industry and our very lifestyle has been releasing into the environment vast amounts of compounds that deteriorate our environment. And it is wrong to think that Mother Nature will solve the problem just by itself. We really have to do something about it because the bacteria that inhabit our planet has not had enough evolutionary time to deal with this immense collection of new chemicals. So by using the knowledge that we have generated over the years in molecular microbiology and environmental biotechnology, we may be able to convert this knowledge into strategies and interventions in the environment that can help solving the problem that we have created ourselves, because nature by itself will be unable to do it. And this is where synthetic biology comes in. So synthetic biology offers a tremendous opportunity to face problems that otherwise will never go by themselves.